Welcome to this last video lecture in our section on membrane structure, membrane transport, and today we're going to get a little bit into some cell junctions. But before we get there, I'm going to talk about a different way that material can enter and exit the cell. Previously we've been talking about moving materials directly across the cell membrane, either by passive diffusion, or by simple diffusion, or by facilitated diffusion, or by active transport. So here is another way, two other ways that materials can move into or out of cells. And things here aren't actually moving across a membrane in the way that we've been thinking about membranes before. A little while ago we talked about the movement of material from the ER to the Golgi to the cell membrane and out of the cell. And that process of moving a vesicle's contents outside of the cell is called exocytosis. Cytos means closed vessel and exo means outside or exterior. So this literally means to move something exterior to the cell. So what's happening here is that we're over on the Golgi on the left, and some proteins or some molecules are going to be packaged into a little vesicle. So what happens here is if this is the kind of one of the lamellae, one of the plates of the Golgi, and inside there are a bunch of molecules that need to be released outside the cell, a small piece of membrane will pinch off of the Golgi so that inside there are the components which need to be released. And that vesicle can move through the cytoplasm, and as it does so there are a number of structural changes we're not going to worry about. And then when the membrane, imagine here is a membrane, this is the outside of the cell, so imagine this is the outside of the cell. As the membrane approaches the cell surface, a bunch of proteins can facilitate the fusion of the vesicle membrane with the cell membrane such that the contents are released outside the cell, and that's called exocytosis. Now actually nothing moved across a membrane here because things were enclosed within a membrane and then released. So nothing's moved across membranes. Things have moved from inside the cell to outside the cell in a little bit of a paradoxical way because they never actually crossed a membrane, unless you want to go back to thinking about how those things got into the Golgi, but that's a bit of a different discussion. Now that process can also be reversed as well, and so the reverse of that process is called endocytosis. Cytos still meaning closed vessel, and then endo meaning within or inside. So endocytosis is what we're going to do is we're going to imagine this is the cell surface. We're going to take a piece of membrane and pinch it off and bring it inside the cell. So the way that happens is that on the cell surface there are receptor proteins which will bind to molecules outside the cell. So here's a cell surface receptor, here's the membrane, here's a cell surface receptor, and a ligand, a molecule can bind to that receptor like is happening over here, the receptor in blue, and so the receptor here binds to the ligand, and then what happens is a small piece of membrane can be pinched off and brought inside the cell. And so when you pinch that off, you bring the membrane into the cell cytoplasm, and anything that was bound to the receptors is now inside the vesicle. And then that can go off, and for example the vesicle could fuse with a lysosome, and whatever was attached to the receptors can be broken down and then recycled back into the cytoplasm. So that is endocytosis. There's another version of this called pinocytosis, and Pinot means small sips. If you ever drank Pinot Noir or Pinot Grigio wine, Pinot means small sips. And so pinocytosis is when the cell is actually sampling small volumes of fluid using this kind of mechanism, but it's fluid that's being brought in rather than receptors with attached cargo molecules called ligands. And that's pinocytosis. It's a form of cell drinking, as what a lot of people call it. Okay, the last thing to talk about here is one little section on junctions. Now, we're not going to talk about a lot of types of junctions. I'm going to leave that to cell biology at the 3000 level. I'm going to talk about one type of junction, the gap junction. The gap junction is a structure which joins two cells' cytoplasms together directly, so that solutes don't have to pass across one membrane and then into a second membrane to get into an adjacent cell. So if there's a cell here and there's a cell over here that needs a solute being made in this cell, what you'd expect to happen is that the solutes leave one cell and then they enter the next cell in two steps, in two sets of processes. Well, there are junctions. These are protein molecules which entirely span the two membranes of the two cells, and they form a little channel. Now these aren't really channels, but they look just like channels, and that means that solutes can move backwards and forwards down concentration gradients by simple diffusion, by diffusion through the channels here. And so these are called gap junctions. And so here over on the left we've got a couple of cells. Here's one cell, then there's another one over on the left and one on the right. And these gap junctions down here are basically allowing materials to move bidirectionally from one cell cytoplasm to another. So these are very complicated protein structures which are joining two cells cytoplasm so things can move back and forth, and they're made from these structures called connexons that basically link up two cells and form this junction so things can move backwards and forwards. So as we're finishing up there, here comes Henry to wish us farewell. Don't forget that the learning outcomes cover everything that you need to know in terms of quiz questions for your quizzes. So we'll finish up there, and Henry and I will bid you a fond farewell, and we will see you next time when we start our next module. So bye for now.